Okay, very good evening, my dear friends. So today's our eminent resource person, Dr.O.Padmaja Madam Garu, Assistant Professor, Department of Botany, Tara Government Degree and PG College, Sangareddy, Autonomous. Madam, please welcome. And Madam has huge experience regarding of in this plant sciences. So Madam is very enthusiastically accepted our request for the services, rendering of services towards the success of the aspirants, those who are going to attend CPGET 2023 and also JL Botany and also DL Botany and Gurukula JL and Gurukula DL and PGT also. So all competitive examinations, aspirants, please, my humble request and suggestion, please utilize this wonderful opportunity. Madam has huge experience regarding of these classes. Madam, please welcome and share your screen and start your session. Madam, please welcome.
Okay, thank you, Tirupati sir, for giving this opportunity. And I felt this will be a good opportunity and a good platform for me, so that the number of students will be benefited for this higher education. And at the same time, number of posts are there in coming years. The number of students will be benefited. Thank you very much, sir. Sir, is it visible, sir?
Yes, it's visible, madam. Please click on slideshow if possible. పైన ఉంది చూడండి, పైన ఈ యానిమేషన్స్ పక్కన ఉంది చూడండి, స్లైడ్ షో పైన యానిమేషన్ పక్కన. రైట్, ఇక్కడ ఫ్రమ్ ది బిగినింగ్. yes clearly visible, madam. Please you start.
Okay sir, thank you sir. Myself Dr.Padmaja, Assistant Professor of Botany. Today I am going to start my topic regarding the embryology, a very short and interesting topic in total syllabus of the botany. So number of branches are there in the botany. Those who are preparing for the JL or a DL or for the higher education, for the interesting to do the PG entrance, this topic is very much helpful for you. So today I'm going to start my topic on embryology. So what are the contents? Means introduction to embryology, and what is the organ, what the embryology, where this organ, which part of the organ of a plant this embryology is going to start, regarding that, and Microsporangium, Microsporogenesis, and development of microspores, and development of male Gametophyte, and conclusion and the question. This is the content I am going to start for this session. Before that, let's enter into the introduction to embryology. Here, embryology is the study of structure of development of embryo, and where this embryo and where the formation of the fruit is taking place is the main part is the flower. So flower is considered as a reproductive part of a plant. So number of life processes are taking place in a plant. For example, photosynthesis or respiration, and like this the reproduction. So if we thought about the photosynthesis, the photosynthesis taking place in the leaf organ in the chloroplast. And if you take the respiration in the mitochondria, like this, the reproduction is taking place in the reproductive part that is known as the flower. So it is also considered as the this flower is helpful for the formation of the fruit, and this fruit for the development of embryo. Just you imagine if a plant is not having the flower, so there won't be any food formation for us also. So this our topic, including the structure and development of male and female reproductive organs and the fertilization and the similar other process. So before entering into that, and who is the person who is considered as the father of Indian plant embryology is Panchanan Maheshwari. And number of research work is did in embryology. The person is Panchanan Maheshwari. He is studied about after that this embryology leads to the tissue culture, and this is leads to the formation of protoplast culture. All these are come after the development of this embryology. And now the plant embryogenesis. The formation of the embryo is the process that produce after the fertilization of an ovule to produce a fully developed plant embryo. This is a patent stage in the plant life cycle that is followed by the dormancy and the germination. See, if you think that if the embryo is in a dormancy condition, it is not having the ability to produce the seedling. So these all we are studied in future in this regarding this embryology.
Ma'am, good evening, ma'am.
Good evening, students. And you already studied that in a plants, there is a division of from lower plants to the higher plants. For example, the plants, the lowermost plants are considered as the algae, fungi, the Bryophyta, Pteridophyta, and the next one is the gymnosperms and angiosperms. In this, the angiosperms is considered as the well-developed plants in the plant society. So in this angiosperm, the reproduction is well developed when compared to the lower plants like the algae and the
Madam, 1 second. Slide is not movable, madam. Only introduction to embryology slide is. Okay, right.
Now we are going to the reproductive organ, that is the part of a flower. So whatever we are going to study regarding this embryology, this is related to the beautiful part of a plant, that is the flower. So the basic, when we consider the basic part of the embryology is the study of the parts of a flower. You already know that the essential organs are there and the non-essential organs are there, and the non-essential organs regarding as the calyx and the corolla, and this is very, very essential in the study of the taxonomy also. And if you see here a beautiful structure, here you already familiar with these all the parts, but we are going to study the depth of this regarding the male gametophyte, regarding the female gametophyte. The future topics will be related to the internal study of this flower only. And if you see here the parts like considered as the corolla, calyx, and the stamen. In the taxonomical word, we consider it as an andrecium, and the taxonomical terminology, this pistil is considered as a gynoecium, and the receptacle and the peduncle, these are the parts of a flower. So the stalk of a flower is known as the pedicel, and the cushion-like structure where all the essential organs and the non-essential organs are joined, this is called as the receptacle. And here the non-essential organs like the sepals are also called as the calyx, and the corolla, which are the beautiful and attractive part of a flower. So this attractive part of a flower is also helpful for the attracting the agents like the insects or some of the even bats or animals for the pollination. So these are considered as the non-essential organs, and the non-essential organs. Now it is we are going to entering into the essential organs, that is the stamen, andrecium, and the gynoecium. So if you see the structure of stamen. It consisting of a strong elongated structure known as the filament, and this is the anther, and the tip is called as the anther. And we may get some bits like regarding that, which type of attachment of this filament to the anther helpful further. For example, if they take the family Graminaceae, the Graminaceae కుటుంబానికి చెందిన ఈ మొక్కలలో అన్నమాట పోయేసి the filament that is attached to the anther is just like a versatile. A tip of this anther is attached to the filament is attached to the anther so that the anther can rotate overall for the disposal of these pollen grains also. So like this, we can also get some type of bits. And next one is this is the center part. It is the gynoecium. So the stalk, that the gynoecium part is known as the the solen part is known as the ovary, and the elongated part is the style, and this is the stigma. And all this process we are going to study now. So already we studied this here, the anther. So this anther, we also come across these. They are dithecous anthers and monothecous anthers. First, you see when we transfer section of these anthers will be, when we cut this transfer section of an anther, it consisting of 4 lobes. And this 2 lobe, this 4 lobed structure is also called as the 4 microsporangia are present in that 4 lobes. And 2 lobes considered as 1 theca and 2 lobes considered as the 2 thecas. So, for example, this dithecous, all the families consisting of dithecous anthers, but this in Malvaceae family, a monothecous anther is present. And when we see the cross section of these anthers, like this, we are going to. This is the structure of this anther. These are the 4 lobes. And this one is called as the 2 lobes considered as the microsporangium. And these are considered as the microsporangium. If we see, this is known as the internal structure of an anther wall. So why we are going to study this means where the pollen grains are present, how the pollen grains are formed. Here the flower is it is a diploid condition, but the pollen grains are in haploid condition. And what is the process that is taking place? The diploid condition is giving rise to the haploid condition. If you see the transfer section of an anther, we may expect some bits from this. The transfer section of the anther, this is the diagram. This is the outermost layer is known as the epidermis. And this below this epidermis is the endothecium. And this below endothecium, there is the middle layer, and a below the middle layer, there is the tapetum. And inside the tapetum, the fertile tissue or nothing but the microspore mother cells are present. So if we see the transfer section of these, the anthers, these are the types of layers we are seeing in the transfer section of the anther. So it is the outermost layer. Below this epidermis, elongated cells known as the endothecium. These are consisting of radially fibers are also present. Below this endothecium, a 1 or 2 layered middle layer will be present, and below that a nutritive layer that is known as the tapetum. This tapetum is helpful for giving food materials for developing microspore mother cells. And whenever these microspore mothers are going to develop, the tapetum loses its identity. And whenever these pollen grains are formed, like this, the rupturing of this anther is taking place at the stomium place. And we also studied that they may be dehiscence. The dehiscence of the anthers will be of longitudinal dehiscence, or maybe porous dehiscence, or maybe the transverse dehiscence. అంటే ఏంటంటే యాంథర్స్ పగలడం అనమాట నిలువుగా పగలడం కానీ లేకపోతే పోరస్ లాగా పగలడం కానీ మనం చూస్తుంటాం. సో at the point of stomium, these microspore mother cells give rise to the pollen grains. So this is the brief idea regarding the formation of the pollen grains. Now we are going to study that the endothecium, middle layer, tapetum, and how this they loses its identity, the this middle layer will, how this loses its identity, like this we are going to see here. So here this the tapetum, so it is considered as the innermost layer, and it is the layer which is surrounding the each microsporangium. This one is called as the microsporangium. The one of the layer is totally rounding that microspore mother cell is the tapetum, and the cells are multinucleated and the polyploid. The number will be, the chromosome number will be more, and multinucleate means more number of nucleus will also be present. They contain Ubis bodies, and these Ubis bodies are helpful for secreting the sporopollenin that is helpful for the insects to attract the pollen grains. And the main function of this is the nourishment. So here when we study the what are the four layers, one we already studied that is the epidermis, endothelium, middle layer, and the tapetum. We can also expect one question from this the regarding the tapetum. So it is an outermost protective layer of the anther wall, and it is made up of tangentially elongated and flattened cells. The epidermis performs its usual protective function. We already know that the outermost layer is helpful for the protecting the internal organs, and the below one is the endothelium. So inner to the epidermis is the endothelium is present. Mostly it is a single layer. Sometimes maybe several layers, like for example the Rathura, and the radial and inner tangential wall of endothelial cells having fibrous thickenings. This is the characteristic feature of this endothelium. So it is also helpful in the dehiscence of the anther wall whenever the pollen grains are mature or the pollen grains are formed from the microspore mother cells by the reduction division. And the next one is the middle layer. Inner to the endothelium, the middle layer is 2 or 3 layers, and due to the pressure of these microspore mother cells, they loses its identity, and they are generally epimeral. Epimeral means they will stay for some time, and when the duty is completed, they will be loses. Like this, they are called, the word is known as the epimeral. They distinguish or crushed. Due to this pressure formed, due to the formation of the pollen grains, due to the pressure, the middle layer will lose its identity. So we already come across this tapetum before only. The innermost layer of anther wall is the tapetum, and it surrounds the sporogenous tissue. So what are the fertile male producing cells that are surrounded in the tapetum? So that sporogenous tissue give rise to the microspore mother cells. These microspore mother cells in turn give rise to the microspores, nothing but the pollen grains. So it serves as a nutritive tissue. Idanamata perugutunna microspore pollen grains ki food ga panjeyadam jarugutundi annamata tapetum. So there are different two types of tapetums. Maybe one is amoeboid tapetum, another one is the secretory tapetum. So amoeboid tapetum, it is also known as plasmodial tapetum. In this type, inner and radial walls of tapetum cells break down early due to the action of hydrolytic enzymes. So why it is called as the plasmodial tapetum? Plasmodial tapetum means due to the wall rupture, all will be present at a one place. They are just like looking like a amoeba. For this amoeba, there won't be any shape like that. They all present in that shape. So it is called as the amoeboid tapetum. Now the protoplast of these cells enter into the anther locules and fuse to form a common mass called the periplasmodium. Type of tapetum will be present. It is considered to be the most primitive one, and it is present in the monocots or the lower dicots. We already know that in the plants, they are different from the primitive to the higher level. If you take the primitive in angiosperms, in the angiosperm there may be monocots and the dicots. So monocots is considered as the primitive one when compared to the dicots. So this type of amoeboid tapetum is present in the monocots. Example, Gutamopsis. And the secretory tapetum. It is also known as the glandular tapetum. It is found in the most of the angiosperms. The tapetum retains the original position and cellular integrity and nourishes the development of the microspores. That means here whenever the tapetum will be due to this enzymatic action, the dissolving the wall and forming a plasmodium-like structure, that is considered as the plasmodial tapetum. And here it is a granular type that's called as the secretory tapetum. Like this, what is the main function of the tapetum is to provide food for the developing microspores. And the nutrients are transported to the tapetum, to the sporogenous tissue. So the tapetum is involved in the synthesis of callose, which is release microspores in tetrad by degrading the callose wall. And it is also helpful for all the pollen grains. They may be in the form of a tetrads. In order to separate, these are also playing an important role. And this tapetum plays an important role in the formation of exine. Exine means the outermost layer of the pollen grain is known as the exine. In sometimes they will give the question like that: what is the role of the tapetum? We already know that is the main function is the nutrition, and it is also helpful in the formation of exine, and it is also helpful for the synthesis of callose. And next one is they give like all. So we have to know the if we know all the points, we can easily put the answer like that. And here the main important is the oily layer present on the surface of pollen grains is contributed by the tapetum. This is called as the pollen kit. These pollen kit are mainly seen in the entomophilous pollinating agents. In that type of pollen grains, we'll have in seeing that pollen kits. So it may act as an insect attractant. It protects the pollen from the UV radiation. So these ubisk bodies is also playing a very, very important role. And now, after that, the study of the transverse section of the anther. ఇప్పుడు మనం ట్రాన్స్ఫర్ సెక్షన్ ఆఫ్ ద యాంథర్ చూసాము. అందులో ట్రాన్స్ఫర్ సెక్షన్ ఆఫ్ యాంథర్ లో ఏమేమి పార్ట్స్ ఉన్నాయి అనుకున్నాము. అండ్ ఈ యాంథర్ లో నుంచి మనకి ఏం వస్తున్నాయి అనుకుంటే ఏంటిది అంటే దీస్ మైక్రోస్పోర్ మదర్ సెల్స్ ఆర్ ప్రెసెంట్. These microspore mother cells give rise to microspore. So tapetum, inside the tapetum there is a tissue that is known as the sporogenous tissue. This sporogenous tissue is a diploid one, and this sporogenous tissue undergoes meiotic division. As a result, 4 microspores are formed from each microspore mother cells. These are arranged in a form of a tetrad. And these microspores are haploid in condition. And these microspores are also called as pollen grains. So the formation of pollen grains from microspore mother cell is known as microsporogenesis. So genesis means giving birth. Microspore mother cells nunchi the microspores are coming. So this is called as microsporogenesis, and these microsporogenesis are taking place in the anther itself. So here you see the formation and differentiation of microspore is called as microsporogenesis. That means the microspore mother cell give rise to microspores. Each microspore is diploid in condition. This diploid microspore undergoes reduction division. As a result, 4 microspores are formed. For example, if the microspore mother cells are
అమ్మవారి దగ్గర ఇక్కడ చూసిన తర్వాత అక్కడ కూడా వెలిగిస్తావు కదా, అక్కడ పెట్టేసి చెట్లు కాకుండా
microsporogenesis. And now, like this, the microspores are formed here. The 4 microspores are in the arrangement of microspores. We also come across that microspore mother cell undergo reduction division. As a result, 4 microspores are formed. And the arrangement, how the arrangement of these microspores are there is, the first one is known as the tetrahedral. Tetrahedral means you already know the methane, the C4. How it will be the 3 hydrogen మనకు కనిపించే ఒక హైడ్రోజన్ వెనకాల ఉన్నట్టుగా ఇక్కడ కూడా మనకి ఏంటి అంటే the 3 microspores are towards one side and one of the microspores are behind. So that arrangement is called as the tetrahedral. In these 4 microspores are present. and isobilateral. Isobilateral arrangement, here upper two microspores and the lower two microspores. And decussate type. Decussate, in this the one towards this side and the one will be the back side. And here this is the T-shaped. T-shaped in this up to, and the two will be down. It is looking like a T. And this is the linear, just like a line. So it like this, the microspores are arranged in this manner. So this is the arrangement of microspores in this formation. They may be tetrahedral, isobilateral, decussate, and T-shaped, and the linear one. And now we're going to see the development of male gametophyte. So the total, whatever we are seeing in the slide, is a very, very important one, and it will be covering the bits also, and it is known as the development of male gametophyte. So what is mean by male gametophyte? Whenever the pollen grains, so the first cell of male gametophyte is pollen grain. Manaki male gametophyte development kavali ante the first cell is the pollen grain. Where the pollen grains are coming from the microspore mother cells. Where these microspore mother cells are there, they are present in the sporogenous tissue. The sporogenous tissue is present in the anther. These anthers are present on the filaments. These filaments or the stamens are present on the flower. So all these are present internally in a flower. So the first cell of male gametophyte is pollen grain. So the pollen grain, it is round in shape, and in some members the pollen grain will be a oval shape in structure, or some may be a spinous like structure, and some may be of showing a ornamental the outside. So the study of pollen grains is known as the palynology. So number of research work are also taking place on this palynology, and even the these pollen grains are also helpful for identifying the criminals, like that in forensics also this research work is going on the pollen grains. So the first cell of the male gametophyte is the pollen grain. It is haploid. Why it is haploid? Because microspore mother cell undergone reduction division. As a result, 4 microspores are formed, or the 4 pollen grains are formed. They are in haploid in condition, and a haploid nucleus is present. Some amount of cytoplasm is present, and it's surrounded by the 2 layers. That is the outer one is known as the exile, and the inner one is known as the intine. And here the exile is not continuous. He is leaving some pores. They are called as the germ pore. And what is the function of the germ pore? These germ pore helpful for the elongation of this entire intine to form a pollen tube. So in some pollen grains, one pollen tube will be formed. That condition is called as the monosiphonous. In some member, many pollen tubes are formed. This is called the polysiphonous condition. And here this is called as the haploid nucleus. This haploid nucleus undergo mitotic division, unequal mitotic division. As a result, 2 cells are formed, one big cell and one small cell. This big one is known as the vegetative cell, and the small one, this is the mitotic division. So unequal division, mitotic division, and the big one is known as the vegetative cell, and the small one is known as the generative cell. And this, after some time, the generative cell will divide to give rise to here. The generative cell will be elongated and a constriction is formed. As a result, the 2 nucleus are separated. These 2 nucleus are give rise to male gametes. these male gametes at this three-celled stage, the pollen grains are released, and these pollen grains are fall. And from the agents like they may be by the wind, or maybe by the water, or maybe by the insects, or maybe by the any pollinating agents. They fall on the stigma of a flower. And as soon as they're falling on the stigma of a flower, the pollen tube will be extending, and this entering from the stigma to the style to the ovary. Like this, the pollen tube formation will be taking place. So the formation of pollen tube from the pollen grain, the total journey is called as the development of male gametophyte. And here, in this three-celled stage, they fall on the stigma, and these are called as the male gametes. And after some time, these will be the male gametes, and the intine will be elongated, and it will be producing this pollen tube. This pollen tube, and these may be elongated, and the tube nucleus is helpful for the showing helpful for the formation of this pollen tube, and it is also helpful for the passage of these micro gametes, or called as the male gametes, for the fertilization process. And after this falling on these two male gametes, the two male gametes is this one male gamete is participating, these two male gametes are participating in the fertilization. So this in angiosperm, the fertilization is considered as the double fertilization. And here these gametes, the male gametes, one of the male gamete will be fuses with the egg, and another male gamete will fuses with the secondary nucleus that are present in the ovule. Like this, double fertilization will be taking place. So this is the study of development of male gametophyte. And from this, from the reproductive part of a flower, we entering into the transfer section of the anther. From this anther, we are entering into the microsporogenesis. And from this microsporogenesis, we are entering into the development of male gametophyte. So in this male gametophyte, number of stages are taking place. In this, the first cell of the male gametophyte is known as the pollen grain. This pollen grain is also called as the microspore, and this pollen grain consisting of this one exine and the intine, and this exine will be of somewhat will be spinous-like structure in some spaces. These spines in hibiscus, if you see these microspores under the microspores under the microscope, it will be looking like a spinous-like structure. And if you see the pollen grains in the Graminaceae, in Graminaceae, a more number of pollen grains are forming because the flowers are very small. So a huge amount of these pollen grains are forming in this. And in some members, these pollen grains are also aggregate to form a group that is called as the pollinia. That pollinia is seen in Asclepiadaceae members and Orchidaceae members. And here, so here this is a spindle-like, the spindle-like structure is the generative cell, and this generative cell later moving towards the center, and it is dividing to give rise to the two nuclei. These two nuclei are called as the male gametes, and these male gametes are, we also studying that if the pollen grains in order to know the viability test of the pollen grains, we will see that the pollen grains, if the pollen tubes are produced, the pollen grains are having the viability test. If the pollen tubes are not formed, they are not having the capacity of the viability. So we can also study by an experiment by the germination of the pollen grains. We can easily study the pollen tube, the length of the pollen tube we can also study it under the microscope by collecting the flower and taking the pollen grains from the anthers, and that pollen grains will be stored in a sucrose solution, and after some time, if we observe under the microscope, we can clearly see the formation of the pollen tubes from the pollen grains. So it is one type of study. For if you want to do a small projects like that, the study of the length, how much the length of the pollen tube is forming, like this, we can also gather the some type of pollen grains from the different families and studying the pollen tube length also. So and the next, development of male gametophyte. So how the process are taking place? The total diagram is telling that about the development of male gametophyte. So first cell of the male gametophyte is pollen grain, and the pollen grain give rise to the pollen tube. So this pollen tube, whenever the pollen tube is formed, the male gametes will be entering into the pollen tubes, and they may be fuses with this egg, and fuses with the secondary nucleus. So the pollen tube containing the male gametes is called as the male gametophyte. And microspores or the pollen grains. So these pollen grains is are haploid in condition. The chromosome number is N, and these pollen grains are also called as the microspores. So it is the first cell of the male gametophyte. The study of pollen grains is known as the Pollenology. And after its release from the anther, the nucleus of the pollen grains undergoes the first mitotic division to form the two unequal cells. In this unequal cell, one will be the vegetative cell, and another, the larger one is the vegetative cell, and the smaller one is known as the generative cell. This generative cell again divides to give rise to the two nucleus. So these two nucleus are called as the male gametes. They are also called as the sperms or the male male gametes were also called as the male nucleus or the male gametes. These male gametes will be fuses with in fertilization. During fertilization, these pollen grains are fall on the stigma of the flower. The stigma is having some sticky nature, and these honey type of structures are helpful for the attracting these pollen grains to fall on that. And some of the there may be some interaction between the pollen grains and the pistil. This is also called as the pollen pistil interaction. Like this, the pollen tube will be forming. This pollen tube will having the male gametes, and the entry of the pollen tube into the ovule may be of depending upon the types. They may be also asking that type of question like the entry, if the entry of the pollen tube through the ovule, micropyle of the ovule, that condition is called as a porogamy. And if the pollen tube entry is towards the integuments of the ovule, it is considered as the mesogamy. If the pollen tube entry is towards the chalaza, that is called as the chalazagamy. So the entry of the pollen tube, whatever may be, that the main function of this pollen tube is to release the male gametes into the embryo sac. What the embryo sac consisting of this, when we study next class, when we study the development of Megasporangium, development of Microsporogenesis, development of female gametophyte, we can come across this embryo sac. This embryo sac consisting of the egg nucleus and the polar nucleus. And one of the male gamete will fuses with the egg nucleus. As a result, zygote will be formed. This zygote will be developing into the embryo. So the difference between zygote and embryo is the zygote is a diploid structure. Whenever the divisions are taking place, that is considered as a embryo. This embryo later on developing into the seed. Like this, the life process in the angiosperms are taking place. So in the next class, we will continue the female reproductive part and the ovule and Microsporangium, Microsporogenesis, fertilization in next class.
Madam, once again, epimerals.
Epimerals means with this type of epimeral word, we also మనం ఇంకెక్కడ వింటాము అంటే, for example, in xerophytic plants. In xerophytic అంటే మీకు కొంచెం ఐడియా కోసం చెప్తున్నాను. కొన్ని ప్లాంట్స్ అన్నమాట ఎండకి తట్టుకోగలుగుతాయి. వాటి ట్రూ జీరోఫైట్స్ అంటాం. కొన్ని ఏం చేస్తాయి అంటే రెస్ట్రిక్షన్ అంటే ఏంటిది అని అంటే, కొన్ని ఎండకి తట్టుకోలేక ఏం చేస్తాయి అంటే ఆ టైంలో అవి కొన్ని ఏమో విత్తనాల రూపంలో ఉండడం జరుగుతాయి. కొన్ని ఏం చేస్తాయి అంటే ఎపిమిరల్స్ అంటే ఏంటంటే దే విల్ ఎస్కేప్, వెళ్ళిపోవడం జరుగుతూ ఉంటది అనమాట. దిస్ ద వర్డ్ ఎపిమిరల్ అనేది యూజ్ చేసే వర్డ్ టర్మినాలజీ, దానికోసం మనం యూజ్ చేస్తాం అనమాట.
అల్పకాలికాలు అంటారు. The entire life cycle within 1 week or 2 weeks, the plant life cycle will going to complete. So plants we can call as epimerals. Some epimerals are tribulus. Tribulus plants. Okay, right? Okay, madam, thank you. Thank you very much. So your valuable time for aspirants.
Sir, first of all, I have to thank for the students. They are very much interested. Interested in the Sunday also they are joining. For the students. Thank you, Tirupati sir.
Thank you, madam. Thank you very much.
Thank you, sir.
Tomorrow, you're coming maybe the same time or in the morning or afternoon?
Sir, from 12 o'clock I will take, sir. Tomorrow
Sure.
at 12 o'clock.
Yes, madam. Yes. Thank you.
Thank you, sir. Thank you.
Thank you very much.